In this chapter, we are going to deal with genetics. Genetics deals with the molecular structure and function of genes, gene behavior in context of cell or organism, patterns of inheritance from parent to the offspring, and gene distribution, variation, and change in population such as through genome-wide association studies. So given that genes are universal to living organisms, genetics can be applied to the study of all living systems from viruses and bacteria through plants and domestic animals to humans. So in genetics, chromosomes plays an important role as its chromosome is an organized structure of DNA and protein found in cells. It is a single piece of coiled DNA containing many genes, regulatory elements, and other nucleotide sequences. Chromosomes also contain DNA-bound proteins which serve to package the DNA and control its functions. In eukaryotes, nuclear chromosomes are packaged by proteins into a condensed structure called chromatin. So this allows the very long DNA molecules to fit into the cell nucleus. Chromosomes are essential unit for cellular division and must be replicated, divided and passed successfully to their daughter cells as to ensure the genetic diversity and survival of their progeny. So chromosomes in humans can be divided into two types, that is autosomes and sex chromosomes. So autosomes, if autosomes are, sex chromosomes contain the genetic traits that are linked to a person's sex. So that's why it is called sex chromosomes. So all other, the rest of the genes uh, contain, or the autosomes that contain the rest of the genetic hereditary information is called autosomes. So two types of chromosomes are present in humans. One is autosome and another is sex chromosome. So you have to be clear about the differences. What determines the sex of an human in humans? So the human sex is predetermined in the sperm gamete. The egg gamete mother cell is said to be homogametic because all its cells possess the double X sex chromosomes. Sperm gametes are deemed heterogametic because around half of them contain the X chromosome and the other half possess the Y chromosomes. In complement that the first X from, in light of this, there are two possibilities that can occur during fertilization between male and female gametes, either double X or X and Y. Chromosomes X and Y, they do not truly make up a homologous pair. They act similarly in their roles, but they are not homologous. They cannot be called as homologous. So the X chromosome in humans is much longer than the Y chromosomes and also contains many more genes. So these genes are said to be sex linked due to the fact that they present in one of the sex chromosomes. So at the time of fertilization, when the opposing homologous chromosomes come together, the smaller Y chromosome offers no dominance against the extra X chromosomes. Let us go to the next topic, that is cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. So cell division is usually a small segment of a larger cell cycle. So this type of cell division in eukaryotes is known as mitosis and leaves the daughter cell capable of dividing again. The corresponding sort of cell division in prokaryotes is known as binary fission. In another type of cell division present in eukaryotes is called as meiosis. It's a cell is in which a cell is permanently transformed into a gamete and may not be divided again until fertilization. Right before the parent cell splits, it undergoes RNA replication. For simple unicellular organisms such as amoeba, one cell division is equivalent to reproduction and an entire new organism is created. So two different cell divisions will be dealing in this one more detail, that is mitosis and meiosis. Mitosis is also known as karyokinesis. It's a division of the nucleus and its chromosomes. So it's followed by a division of the cytoplasm known as cytokinesis. Both mitosis and cytokinesis are part of of the life of a cell, or called the cell cycle. Most of the life of a cell is spent in non-dividing phase called as interphase. Mitosis can be divided into many phases like interphase, prophase, metaphase, anaphase. Before dividing, a cell enters mitosis, it undergoes a period of growth, or called that growth phase is called as interphase. So some 90% of the cell's time in the normal cellular cycle is usually spent in interphase. So the interphase is interdivided into three stages, that is G1 phase, in which the synthesis of DNA. It's called the period prior to the DNA synthesis. So in this, G1 phase, the cell increases in mass in preparation for the cell division. That the cell preparation is started before the cell division. So S phase, it is the period where the DNA synthesis or the DNA is synthesized. G2 phase, the period after the DNA synthesis has occurred, but prior to the start of the prophase. The cell synthesizes proteins and continues to increase in size in the G2 phase. Like in the later part of the interphase, the cell still has nuclei present within it. So the nucleus is bounded by a nuclear envelope, and the cell's chromosome have duplicated, but not in the form of chromatin. So the first phase of mitosis can be called as a prophase. The interphase is just the beginning, or just an interface between the mitosis and the prophase. In prophase, the chromatin condenses into discrete chromosomes, and the nuclear envelope breaks down and spindles form at opposite poles of the cell. That is what happens in prophase. Uh, from prophase, when it enters the metaphase, the spindle fully develops and the chromosomes align at the metaphase plate. Equal metaphase plate means it's a plane that is equally distant from the two spindle poles. That is called as metaphase plate. And here the nuclear membrane, with the where the interface in the nuclear membrane was present in the prophase, the nuclear membrane disappears completely in the metaphase. Then the cell goes to the next stage, that is anaphase. So in anaphase, the paired chromosomes, that is sister chromatids, separate and begin moving to the opposite ends or poles of the cell. Here the spindle fibers are not connected to chromatid lines and elongate the cell. So at the end of the anaphase, each pole contains a complete compilation of the chromosomes. In preparation for the telophase, the two cells of the poles also move further apart during the course of anaphase, and at the end of anaphase, each pole contains a complete compilation of the chromosomes, as I said before. So telophase, the last phase of the mitosis. In telophase, the chromosomes are corded off into the distinct new nuclei and emerging daughter cells. That is, many changes occur in the telophase. That is, the polar fibers continue to lengthen. Nuclei begin to form at opposite poles. So nucleoli also reappear. These all changes happens in telophase. Then comes the cytokinesis. It is oftenly misunderstood that it is the fifth part of the mitosis, or it is a next stage. It happens from telophase to cytokinesis, but it's actually a separate process that begins at the same time as telophase. In both animal and plant cells, cell division is also driven by the vesicles derived from Golgi apparatus, which move along microtubules to the middle of the cell. But in the plants, in this structure is coalesces into cell plate, because they doesn't have that microtubules. Plants, it is replaced into cell plate. So at the cell plate, at the center of the cell plate of the and it develops a cell wall separating the two nuclei. So each daughter cell has a complete copy of the genome of its parent cell. So at the end of the cytokinesis, there will be two. You will be receiving two distinct daughter cells. Next division is meiosis. Meiosis is a special type of cell division that occurs in sexually reproducing organisms. Meiosis reduces the chromosome number by half, exactly into half, so enabling the sexual recombination to occur. Meiosis of diploid cells produces haploid daughter cells which may function as gametes. So the stages of meiosis can be broken into two main stages, that is meiosis 1 and meiosis 2. Meiosis 1 separates the homologous chromosomes, producing two haploid cells, that is N chromosomes, that is in human it is 23 number. So meiosis 1 is referred to usually as a reductional division because it is reduced to half. A regular diploid human cell contains 46 chromosomes and is considered 2N because it contains 23 pairs of homologous chromosomes. But in meiosis 2, that is the second part of the division, can be called as an equational division, similar to mitosis, which will occur whereby the sister chromatids are finally split, creating a total of 4 haploid cells, that is 23 chromosomes in, 2 from each daughter cells from the first division. So prophase 1, meiosis 1 starts with the prophase 1. So it might be called as the longest phase in meiosis because during prophase 1, DNA is exchanged between homologous chromosomes in a process called as homologous recombination. So this often results in chromosomal crossover. The new combinations of DNA created during crossover are a significant source of genetic variation and may result in beneficial new combination of alleles. The paired and replicated chromosomes are called bivalents or tetrads. Those have two chromosomes and 4 chromatids with 1 chromosomes coming from each parent. It receives 1 chromosomes from each parent, that from the mother and the father. The process of pairing these homologous chromosomes can be called as a process called synapsis. It plays a very important role in meiosis. So at this stage, when synapsis happens, the non-sister chromatids may cross over at points called as, the points are called as chiasmata. In turn, like prophase 1 is in turn divided into like protein, zygotine, pachytene, and diplotin and diakinesis. Usually, then the diakinesis, the chromosomes condense further, and it forms the first point in meiosis where the 4 parts of tetrads are actually visible to us. So sites of crossing over entangle together, effectively overlapping, making chiasmata clearly visible. Metaphase 1, homologous pairs move together along the metaphase plate. So, as a part of that, microtubules from both centrioles attach to their respective kinetochores. The homologous chromosomes align and along an equatorial plane that bisects the spindle. So, due to continuous counterbalancing forces which exerted on the bivalence by the microtubules, anaphase 1, in that the bipolar spindles microtubules shorten, serving the severing the recombination nodules and pulling homologous chromosomes apart. So since each chromosome has only one functional unit of a pair of kinetochores, the whole chromosomes are pulled toward opposite side or opposite poles, forming two haploid sets. So each chromosome might contain they might still contain a pair of sister chromatids. Telophase 1, the last meiotic phase effectively ends when chromosomes arrive at the pole. So each daughter cell now has half the number of chromosomes because it's an haploid number. But each chromosome consists of a pair of chromatids. The microtubules that make up the spindle network disappear in the telophase 1 and a new nuclear membrane surrounds each haploid cell. So as a result, the chromosomes uncoil back into chromatin. Cytokinesis, that is the pinching of or the vesicles formation cell membrane animal cells or the formation of cell wall in the plants, occurs completing the creation of two daughter cells, that is sister chromatids remain attached to the telophase. At the end of the telophase 1, we will be having two sister chromatids with us. Cells may enter a period of rest known as interkinesis or interphase 2. So, but in this, the no DNA replication doesn't occur at this stage. The next step, that is the equational division, you can say, is the meiosis 2. Meiosis 2, which is also similar to mitosis. So most of the steps are the similar. Most of the changes occurring in mitosis is happening in meiosis 2 also. The end result is that we'll be receiving 4 haploid cells from 2 haploid cells, each 23 chromosomes, each of the chromosomes consisting of two sister chromatids produced in meiosis 1. So altogether, the four main steps of meiosis 2 are usually it's the same as the mitosis. So it's divided into meiosis 2 is divided prophase 2, metaphase 2, anaphase 2, and telophase 2. But in now at the end of the meiosis 2, there is a complete and it ends up with 4 new cells. Mendelian genetics. Mendel has played an important or can be called as the father of genetics. Mendelian inheritance is a study or it's a scientific theory of how hereditary characters are passed from the parent organisms to their offsprings. So the law of inheritance was first derived by Vigor Mendel, a 19th century Austrian priest known conducting hybridization experiments in garden peas. So it was between 1856 and 1862 that he cultivated and tested some 29,000 pea plants. So from those experiments, he deduced that two generalizations which later became as Mendel's principles of heredity or Mendelian inheritance. So Mendel worked on inheritance in sweet peas and selected characters that bred true. That is, the characters were true characters. That characters did not blend into one another in the next generation. So they were true characters. So they were chosen by Mendel that included flower colors such as red, white, plant height versus tall versus dwarf, sweet coats such as smooth coated seeds versus wrinkled seeds. So he eventually formulated three laws of genetics. Now it is known today as the Mendel's law of inheritance. It was in divided into three, like law of segregation, law of independent assortment, and law of dominance. So in law of inheritance, or it was inter subdivided, or it can be the whole three laws together is called as law of inheritance. So law of segregation states that genes segregating during gamete formation into different alleles, that is the two members of a pair of alleles separate or segregate into two different gametes and exert their influence in the offspring as one of a new pair of allele. So segregation is the result of the separate carriage of genes on chromosomes which are not altered or blended by forming pairs. The second law of inheritance, that is the law of independent assortment, said that, or describes that, the chance distribution of alleles to the gametes, or the ova or spermatozoa. For example, if an individual has two pairs of alleles, say capital A small a and capital B small b, so it its gametes will contain equal number of the four possible combinations with one member from each pair. So independent assortment applies only to genes lying on different chromosomes, not on the same chromosome. That is, X-linked chromosome, it doesn't appear. It doesn't apply to the linked genes on the same chromosomes. The third Mendelian law of inheritance, that is law of dominance, states that hereditary factors work together as sets, usually as pair of alleles. So the total number of different alleles represents some of the variation available to species. So frequently in heterozygotes, that is with one dominant and one recessive allele, only the dominant allele of the gene is expressed in the phenotype. The recessive allele being concealed and is usually not expressed. So that gave rise to the Mendel's law of inheritance or law of dominance. Next, we'll deal it with what is sex linkage. Sex linkage is the phenotypic expression of an allele related to the chromosomal sex of the individual. So this mode of inheritance, in contrast to the inheritance of traits on autosomal chromosomes, that is chromosomes which doesn't apply with the, or doesn't carry any sex chromosomes with them. So where both sexes have the same probability of inheritance. So since humans have many more genes on the X than the Y, there are many X-linked traits than Y-linked traits. Usually in mammals, you can see that females is always homozygous sex with two X chromosomes, while the male is heterozygous with one X and one Y chromosome. Morgan has done an experiment to prove this excellent trait excellent inheritance. Uh, he has done this experiment using Drosophila. Pedigree analysis is another important field. It's usually used. Pedigree is it's a diagram of family relationship that uses symbols to represent people and lines to represent genetic relationships. See, these diagrams make it easier to visualize the relationship with their families, particularly the large extent. I mean, if they are, how they are linked to their grandparents, and how what type of diseases the offspring can, or the for F1 generations or F2 generations can are prone to which all diseases, and that can be determined through this pedigree analysis. So pedigrees are often used to determine the mode of inheritance, whether it is dominant or recessive of genetic diseases. genetic diseases and or genetics and human diseases, how they are correlated. Any of the diseases or disorders that are caused by mutations in one or more genes can cause human diseases. So genetic diseases in humans are caused maybe due to the abnormalities in genes or chromosomes. So such defense can be like maybe used because of mutations. What kind of genetic abnormalities can cause diseases? That is, one type is mutations. Mutations are actually sudden inheritable changes in the nucleotide sequence of a gene. So that might cause a different a new disease. Aneuploidy. It is aneuploidy means it's caused when there are abnormal number of chromosomes in an organism. Like from the apart from the normal number, if they have you can see normal number of chromosomes, and this could be due to the loss of a this abnormality may be due to the loss of a chromosome or the presence of extra copy of a chromosome. It might be due to deletions, that is loss of a part of a chromosome, or duplications, that is a portion of chromosome that results in extra amount of genetic material. It might also be due to the inversions of the nucleotide sequence because a portion of chromosome has broken off, got inverted, and reattached at the original location of the chromosome. So all this can happen. So types of genetic disorder. What all types can we find? First one is autosomal dominant genetic disorder. So autosomal dominant genetic disorders are caused when an individual has inherited the defective gene from a single parent. So this defective gene belongs to an autosome, not to an sex chromosome. So such an inheritance is known as autosomal dominant patterns of inheritance. An example can be dwarfism or polydactyly, which in which you will be having an extra finger or toes. Second is autosomal recessive genetic disorder. Such disorders manifest only when an individual has got two defective alleles of the same gene, one from each from the parent. That is, these genetic disorders are inherited via the autosomal recessive pattern of inheritance. That is sickle cell disease or cystic fibrosis. Third one is sex-linked disorders. So these are the disorders related to sex chromosomes or genes in them. That is, one is color blindness and hemophilia. Multifactorial genetic disorders, such diseases are the result of genetics, as well as it might include both maybe genetics or genetic disorders, or it might be environmental factors that are affecting some, such as the heart defects, cleft lip, and cleft palate. All this belongs to like it might be due to some other reasons, some unexplained reasons we can say. So all this might help us to know more about the genetic diseases and disorders, as the scientific study has shown that more genetic disorders are coming up today. These are because using the pedigree analysis they will be able to rectify all these errors. So that is the main I mean, one of the main reasons why pedigree analysis is being used more today to track up the relationships and track up the uh, diseases that the coming generations are prone to.